You can see here the chart prepared by my student on the glycolysis, where the sequence of reaction converting glucose to pyruvate or lactate. And in whole glycolysis there are actually three phases. One is energy investment phase, one is called splitting phase, and the energy generation phase. And there are actually 10 steps in the glycolysis. 3 are irreversible stages and remaining 7 are reversible phases. Now we'll start with the first step. The glucose, we have taken the open chain form of the glucose structure, and here the enzyme glucokinase or hexokinase will act and glucose converted to glucose 6-phosphate. Actually it is the phosphorylation process where 6 carbon containing glucose converted to 6 carbon containing glucose 6-phosphate, and glucokinase act when the glucose concentration is very high, whereas hexokinase act when the concentration of glucose is low, and hexokinase it occur in all the tissues, whereas glucokinase it occur in only the liver. And for this process the coenzyme is needed, from which we have taken the 1-phosphate group, and the glucose, look at the 6 carbon which is converted to glucose 6-phosphate at the carbon number 6, and the process is called as phosphorylation. And here you can see that glucose, which is very permeable to the cell membrane, and we need to restrict that glucose into the cell membrane, and that's why glucose must be converted to glucose 6-phosphate, so it becomes more polar in nature, and the overall glycolysis occur in the cytoplasm. And this step, first step, is a rate limiting step because it is irreversible step. First out of 3, the first step is irreversible. And the kinase are the type of enzyme they cause the phosphorylation type of reaction. Gluco, because glucose is there, kinase for phosphorylation. Hexo means there are 6 carbon are involved, kinase because the phosphorylation occurs. Now that glucose 6-phosphate, which is 6 carbon containing compound, converted to fructose 6-phosphate. Glucose and fructose, they are having the chemical same empirical formula, but there is a structural difference. They are isomers of each other. That's why the reaction is isomerization, and the name of the enzyme is phospho, because phosphate group is there. Hexose, both are hexoses, 6 carbon containing compound. Isomerase, because isomerization from glucose 6-phosphate to fructose 6-phosphate, both are 6 carbon containing isomerization. And now the last step in the first energy investment phase, again we have converted fructose 6-phosphate to fructose 1,6-bisphosphate. So carbon number 6 already contains a phosphate group, and now the phosphate, phosphate is taken from the ATP and it is converted to fructose 1,6-bisphosphate. ATP is taken, that's why from ATP phosphate group is taken, so phosphorylation, and the name of the enzyme is phosphofructokinase. Phosphate group is there, fructose get converted kinase because of the phosphorylation process, and the whole phase is called as energy investment phase. Now the second phase, where the splitting phase, where that fructose 1,6-bisphosphate highly energetic compound get cleaved into 2 3 carbon containing compound. One is called as a DAP and one is called as a GAP, and the enzyme is aldolase, because this is aldolase condensation type of reaction. That's why the name of the enzyme is aldolase, and here the cleavage or splitting occurs. Fructose 1,6-bisphosphate, first 3 carbon, they are converted into dihydroxyacetone phosphate, the first 3 carbon, and the last 3 carbon, you can see here glyceraldehyde 3-phosphate GAP. DAP and GAP both are 3 carbon containing compound, so 6 carbon containing fructose get converted into 3 carbon containing compound, and both are isomers of each other, and the reaction is a reversible type of reaction. So the isomerization takes place. GAP can be converted into DAP or vice versa, and the name of the enzyme is phospho because phosphate group is there, triose because both are 3 carbon containing compound, isomerase because the both can be interconvertible, isomerization can be takes place. And here the BAHP is the inhibitor, which is the competitive inhibitor for this phosphotriose isomerase. So this is about the splitting phase, energy investment, splitting, and now energy get generated via the glycolysis. So we can say one glucose molecule can be converted into 2 GAP molecule, 2 molecules of glyceraldehyde 3-phosphate GAP. Now this glyceraldehyde 3-phosphate then converted into 1,3,1 and 3 both contains the phosphate group 1,3-bisphosphoglycerate. So one more phosphate group is added, and that is from the inorganic phosphate from the cytoplasm. And so here phosphorylation is also there, that is oxidative phosphorylation, and simultaneously there are 2 reactions. One is phosphorylation as well as there is a oxidation reduction type of reaction. That's why the name of the enzyme is glyceraldehyde 3-phosphate dehydrogenase. Dehydrogenase is enzyme there because oxidation type of oxidation reduction reaction is there, and glyceraldehyde 3-phosphate get oxidized to 1,3-bisphosphoglycerate, and simultaneously NAD get reduced to NADH plus H plus. NAD get reduced and our glyceraldehyde get oxidized, and simultaneously phosphorylation is there, oxidative phosphorylation, and our first energy is generated here, the 2 NAD converted to 2 NADH, that is oxidative phosphorylation. That's why via the ETC the ATPs are synthesized, and here the glyceraldehyde 3-phosphate can inhibited by the the inhibitors iodoacetate or arsenate are the inhibitor for this enzyme. Then the next step involves the 1,3-bisphosphoglyceraldehyde can be converted to 3-phosphoglycerate by the enzyme phosphoglycerate kinase. Kinase is the enzyme. Here actually dephosphorylation occurs, that's why the name of the enzyme is kinase, and here our second substrate level ATP is produced, so the ADP converted to ATP, and this enzyme require Mg2 plus as the coenzyme. You can see here 3-phosphoglycerate at the 3rd carbon, phosphate group is there, and dephosphorylation occurs, and here it is substrate level phosphorylation. That means the ATPs are generated without the involvement of the ETC. So 2 NAD converted to 2 NADH, 2 ADP converted to 2 ATP, and here 3-phosphoglycerate, which is 3 carbon containing compound. Now this 3 carbon containing phosphoglycerate converted to 2-phosphoglycerate. The just shifting of that this phosphate group from carbon number 3 to carbon number 2. So it is just a isomerization type of reaction. 3-phosphoglycerate converted to 2-phosphoglycerate by the enzyme phosphoglycerate mutase, just isomers of each other 3 to 2, just interconversion of phosphate group. Now that 2-phosphoglycerate then converted into phosphoenol pyruvate. You can see here the 1,3-bisphosphoglycerate, which is very high energy compound because it contains the 2 phosphate molecules. Now the second highest energy containing compound is phosphoenol pyruvate, and the enzyme is enolase, which convert 2-phosphoglycerate to phosphoenol pyruvate. And here dehydration, the step is dehydration, where water molecule is removed and Mg2 plus are the coenzyme. You can see here the structural changes of the 2-phosphoglycerate to phosphoenol pyruvate, and for the enolase, fluorides are the inhibitor. When we estimate the glucose from the blood, these fluoride molecules are added so that it will stop whole the glycolysis process, and we can easily or exactly determine the amount or concentration of glucose from the blood, and that's why fluoride molecules are added. Now that phosphoenol pyruvate finally converted to pyruvate, and here the third irreversible step, that is pyruvate kinase, which cause the third irreversible or rate limiting step. Pyruvate kinase, because again a substrate level phosphorylation, where again one more ATP is produced from the ADP. That means the phosphate molecule from this phosphoenol pyruvate is removed, and now it is converted to pyruvate, and that phosphate is taken by ADP to convert it to ATP, and the pyruvate, which is 3 carbon containing molecule, is produced, and the enol, which is unstable, can be converted to keto form of the pyruvate. Now this is the end product of the glycolysis when under aerobic condition, and glucose converted to pyruvate. Under anaerobic condition, the glucose finally converted to lactate from the pyruvate by the enzyme lactate dehydrogenase, and energy is invested here for this, and because anaerobic condition, oxygen is absent, and pyruvate converted to lactate. Now you can see here the overall energetics of the glycolysis cycle. You can see how much energy is produced, how much energy is used. So first, in energy generation phase, you can see here we have added invested 2 ATPs, one from glucose to glucose 6-phosphate, and another one is from fructose 6-phosphate to fructose 1,6-bisphosphate. So total number of 2 ATPs we have invested. Now you can see how many ATPs are generated here. The glyceraldehyde 3-phosphate to 1,3-bisphosphoglyceraldehyde, because 2 molecules taken part, so 2 NADH are produced. Then from 1,3-bisphosphoglyceraldehyde to 2,3-phosphoglycerate, again 2 ATPs, substrate level ATPs are generated. Here oxidative phosphorylation, 2 NADH are produced, and the last one where phosphoenol pyruvate here converted to pyruvate, and again 2 ATPs are generated. So you can see here the ATPs formed are 2 NADH, so total number of 6 ATPs. Here 1,3-bisphosphoglycerate to 3-phosphoglycerate, 2 ATPs, and from phosphoenol pyruvate to pyruvate, again 2 ATPs. That means total 10 number of ATPs are generated via the glycolysis, but number of ATPs utilized are 2, so minus 2, 10 minus 2, total 8 ATPs are generated via the glycolysis cycle. And you can see here the overall reaction of the glycolysis. We have invested glucose, 1 glucose molecule, and we have generated the 2 pyruvate molecule. From 1 glucose we generated 2 pyruvate. So 2 inorganic phosphate are added, 2 ADPs and 2 NADH. So 2 ADPs converted to 2 ATPs, and 2 NAD converted to 2 NADH plus 2 plus, and 2 water molecules are removed. So this is the overall glycolysis reaction. So there are 3 phases: energy investment, splitting, energy generation, and there are total 10 steps, out of which 3 are where glucokinase hexokinase act, where phosphofructokinase act, PFK act, and where pyruvate kinase act. These 3 steps are irreversible, and the remaining 7 are reversible steps. So glycolysis means breakdown of glucose to pyruvate and lactate. Okay, so you can give me the feedback. Thank you.